Okay, let's unpack this. Imagine if the food you ate today, maybe your sandwich or that afternoon coffee, wasn't just fueling your body, but was also, deeply intertwined with your risk of cancer,
Hmm.
or even shaping how well treatments might work for you. Sounds a bit like science fiction, doesn't it?
It really does.
But this intricate connection, it's actually far more real and present than, well, than maybe you think. Today, we're taking a deep dive into an incredibly complex, yet utterly fascinating area of science.
Hmm.
We're looking at how your diet, the trillions, literally trillions, of microbes living inside you, and the whole landscape of cancer are all connected.
Yeah.
Our mission here is simple: to give you a shortcut to being truly well-informed. We want to pack this with surprising facts and vital insights that could genuinely change how you view your everyday choices.
And it's fascinating because for a long time we often viewed diet's impact on cancer in isolation, right? We'd ask, is this food good or is that food bad for
Yeah,
cancer?
yeah, simple lists.
Exactly. But what's truly emerged as a game changer is understanding that many of diet's effects are actually mediated by our microbiome.
Okay, mediated, what does that mean exactly?
It means the diet doesn't always act directly on our cells, but it changes the microbes first, and then those microbes influence our cells. So understanding this three-way interaction, diet, microbiome, and us, the host, is proving absolutely crucial for the future of cancer strategies.
So it's like we're not just feeding ourselves.
Exactly. You're feeding an entire internal ecosystem, and that ecosystem, in turn, is constantly talking back to your cells, influencing health and disease.
Okay, so let's start with that foundational connection then. We all kind of know diet affects general health, but how directly does it tie into cancer itself? What are some of the clearer links we've, managed to nail down?
Well, the connection is certainly multifaceted. Directly, we do see some really clear associations. For instance, there was this massive analysis, they called it an umbrella study.
An umbrella study, like it covers everything.
Sort of, yeah. It pulled together data from over 800 comparisons, a huge amount of information, and it showed significant links. Alcohol consumption, for example, is directly tied to increased risks for several cancers: head and neck, esophageal, colorectal, liver, and breast cancers.
That's quite a list.
It is. And then there's what we often call a Western diet. Lots of pre-processed foods, refined grains, red or processed meats.
The typical fast food or convenience diet for many.
Precisely. And that pattern is strongly linked to higher risks for pancreatic, colorectal, ovarian, breast, and prostate cancers. It can even make existing colorectal cancer spread faster, promoting metastasis.
Wow. So it's not just causing it, but potentially making it worse.
That's what the association suggests. Conversely, diets we typically think of as healthy, packed with vegetables, fruits, maybe some dairy, and importantly, fiber. These are consistently associated with a lower prevalence of many cancers.
Okay, that part makes intuitive sense, eat your veggies.
Hmm.
But I've seen this term pop up in the research, reverse causality.
Mm.
And sometimes the data seems, conflicting.
Yeah.
How do we untangle that? It sounds like it could really muddy the waters.
That's a really critical point and something we always have to keep in mind. Many of these associations come from observational studies. That means we see two things happening together, like a Western diet and cancer risk. But it's hard to definitively prove one causes the other based on that alone.
So like, does having cancer maybe make someone change their diet to eat more processed foods because they're tired or unwell?
Exactly. Or does the diet come first and increase the risk? It can be a real chicken and egg scenario. It's difficult to untangle which way the causal arrow points sometimes. And yes, conflicting data absolutely exists. Take soy intake and breast cancer risk, for example. You'll find studies suggesting a protective effect, others suggesting no effect, or even potential risk in specific subgroups. These differences can arise from variations in study design, who the participants were, how long they were followed. It makes giving one single, simple recommendation really challenging.
So it's definitely not always a straight A causes B line. And beyond these, let's say, more direct dietary links, are there indirect ways our diet can kind of set the stage for cancer? Almost like creating a more welcoming environment within our bodies.
Absolutely. Think of it precisely like that, creating a more hospitable environment for cancer to take root and grow. Diet-induced obesity is a major one. It's now recognized as a significant cancer risk factor for, I think, at least 13 different types of cancer.
13, wow. How does obesity do that?
It's complex, but it involves several mechanisms working together. Obesity often leads to chronic low-grade inflammation throughout the body. It disrupts normal hormonal signaling, think insulin, estrogen. It puts stress on cellular machinery like the endoplasmic reticulum.
Hmm.
And crucially for our conversation today, it contributes to what we call a dysbiotic microbiome.
Dysbiotic, meaning unbalanced.
Exactly. An imbalanced internal microbial community, often with fewer beneficial microbes and more potentially harmful ones. All these factors, inflammation, hormones, cell stress, dysbiosis, combine to create an environment where cancer is just more likely to develop and thrive.
Okay, so obesity is like a multi-pronged attack,
Yeah.
creating this pro-cancer environment. What about other indirect links? The outline mentioned cachexia.
Yes, cancer cachexia. This is a really devastating condition. It's severe involuntary weight loss, particularly muscle wasting, that affects many patients with advanced cancer. It significantly impacts quality of life and survival.
Can diet influence that?
It's complicated. While no single diet necessarily causes or worsens cachexia directly, there's research exploring if certain diets can help manage it. Some studies suggest the Mediterranean diet or even a ketogenic diet might show promise in ameliorating cachexia in specific cancer types.
Ketogenic, so very low carb, high fat.
Right. But here's where the nuance comes in again, showing how complex this is. There was a rodent study, for instance, where a ketogenic diet did reduce tumor growth, which sounds good,
Wait.
but it actually accelerated the cachexia, the muscle wasting, and that negatively impacted overall survival in those specific models.
Oh, wow. So it helped fight the tumor but hurt the body in another way.
Exactly. It highlights that an intervention might be beneficial for one aspect of cancer, like tumor growth, but potentially detrimental for another, like body composition or overall survival. It's rarely, if ever, a one-size-fits-all solution.
That dual impact is such an important distinction.
Okay.
Let's do a quick recap for everyone listening. We've established that diet has both direct links to cancer, think alcohol increasing risk for several types, and indirect links, often through conditions like obesity, which creates a pro-cancer environment via inflammation and hormonal changes. But we also have to be careful about reverse causality and conflicting data. It's complex. So thinking about this complexity, what questions does this raise for you about how you view diet advice, especially those simple good food, bad food lists? Now this brings us to the next truly incredible player in this story, our internal ecosystem. Let's talk about the microbiome. You said we're feeding this ecosystem. We are definitely not alone in our bodies, are we?
Oh, far from it. Our mucosal surfaces, especially our gut, are absolutely teeming with trillions of microorganisms, bacteria, fungi, viruses, a whole complex community we call our microbiome.
Trillions. It's hard to even picture that number.
It really is. And the emerging evidence is just overwhelming. This ecosystem plays vital roles in, well, pretty much every step of cancer. From its initial development, how it progresses, even how it spreads or metastasizes. Think of them as tiny, tireless residents inside you, constantly interacting with your cells, your immune system, your environment.
What kind of roles are they playing? Are they like little helpers, or can they be, saboteurs too?
Both, remarkably. They can have genotoxic effects.
Toxic.
Yeah.
Like toxic to our genes.
Precisely. Some microbes produce compounds that can directly damage our DNA. Over time, this damage can lead to mutations, some of which might be cancer-causing.
Wow.
They can also influence how cancer cells signal to each other and to surrounding cells, affecting growth. And crucially, they powerfully regulate the immune environment around a tumor, sometimes helping the immune system fight the cancer, other times, unfortunately, helping the cancer hide from the immune system.
Can you give an example of how we know this?
Sure. A really striking one involves germ-free mice, mice raised in a completely sterile environment with no microbes of their own.
Okay.
Scientists gave these mice fecal microbiome transplants, or FMTs, essentially transferring gut bacteria from donors. Some mice got microbes from healthy human donors, others got microbes from human colorectal cancer patients.
And what happened?
The results were stark. The mice receiving the microbiome from colorectal cancer patients developed significantly more early-stage tumors than the mice receiving microbes from healthy people.
Whoa. So the cancer patient's microbes actually promoted cancer development in the mice.
That's exactly what the study showed. It's strong evidence that the gut microbiome composition itself can directly influence cancer risk. And it's not just local effects in the gut. These gut microbes can also impact cancers far away, like in the liver or even the lungs, by modifying our systemic immune responses, the immune activity throughout the whole body, and by producing thousands of different bioactive compounds called metabolites.
Metabolites, like the byproducts of the microbes, living and eating.
Exactly. Think of it as microbial exhaust, but this exhaust contains thousands of different chemical signals that can travel through our bloodstream and influence cells all over the body, including cancer cells or immune cells. For instance, certain gut bacteria can transform a chemical called BBN into another one called BCPN, and this transformation process seems to contribute to bladder cancer development, at least in mice models.
That's incredible, the remote control aspect. Does the microbiome also influence how well cancer treatments work? Because that would be a huge game changer for patients.
Absolutely. This is one of the hottest areas of research right now. Certain bacteria seem to significantly enhance the efficacy of therapies. For instance, specific gut bacteria like Bacteroides fragilis or Akkermansia mucinifila have been shown in studies, often in mice but increasingly with human correlations, to boost the effect of immunotherapy.
Immunotherapy, that's the treatment that helps our own immune system fight the cancer.
Yes, exactly. Drugs like checkpoint inhibitors. Having the right gut microbes seems to make these drugs work better for some patients.
But you mentioned reverse causality
Ah.
before. Does that apply here too?
Yes, we absolutely have to be cautious. For example, studies might show that patients who receive antibiotics around the time of certain therapies, like CAR T-cell therapy.
RT, that's where they re-engineer a patient's immune cells.
Correct. Patients on antibiotics might have poorer outcomes. But is it because the antibiotics wiped out beneficial microbes needed for the therapy to work? Or is it simply that sicker patients who might have worse outcomes anyway are more likely to need antibiotics for infections?
Right, it's hard to separate those factors.
It is. Disentangling direct microbial effects from other clinical factors is a major ongoing challenge in this research.
And you mentioned the gut microbiome, but this isn't just happening in the gut, is it? This microbial influence is body-wide.
That's right. The oral microbiome, the microbes in our mouth, is implicated in head and neck cancers, particularly oral squamous cell carcinoma. Certain bacteria, like Fusobacterium nucleatum, are often found enriched in those tumor tissues.
Oh.
The vaginal microbiome is linked to gynecologic cancers, like cervical or ovarian cancer. And perhaps even more surprisingly, there's this emerging, though still debated, concept of a low biomass intratumoral microbiome.
Microbes inside the tumor itself, even in places we thought were sterile.
Potentially, yes. The idea is that microbes might actually live within tumor tissues, even in cancers not directly connected to surfaces like the gut or skin. These internal tumor microbes have been suggested to impact tumor growth and even treatment response. For example, some research suggests certain bacteria within pancreatic tumors can metabolize the chemotherapy drug gemcitabine, basically inactivating it before it can kill the cancer cell.
Making the chemo less effective.
Exactly. Now, it's crucial to note there are methodological concerns here. Detecting low amounts of microbes is tricky, and contamination of samples is a real risk. So this area is still under intense investigation. But the concept that microbes might reside within tumors and influence them directly is incredibly powerful.
Okay, let's pause for another quick recap. Our bodies host a vast microbial ecosystem, the microbiome, mainly in the gut but present elsewhere too. These microbes aren't just passive residents. They actively influence cancer at all stages: development, growth, spread through things like DNA damage, cell signaling, and immune modulation. We saw a stark example with the FMT study. They even impact non-gut cancers via metabolites and systemic immunity, and critically, they seem to affect how well treatments like immunotherapy work. There's even evidence, though needing more research, for microbes within tumors. So, considering this huge microbial influence, how does it make you think differently about things like antibiotics or maybe even hygiene practices? So, if our microbiome is such a key player and our diet feeds it, let's connect those dots directly. Our diet is clearly a dominant way we shape these microbial communities, right? How exactly does what we put on our plate change this internal landscape? And once those microbes are altered, what are the knock-on effects for cancer risk?
You're right. Diet is arguably the most powerful day-to-day modulator of our gut microbiome. And dietary changes can alter the microbiome composition incredibly quickly, sometimes within just a few days. It's a very dynamic relationship.
Within days, that's fast.
It can be, yes. The mechanisms driving these changes are varied. Let's break down a few key ways diet talks to the microbes. First, there's direct promotion. Think of it like putting specific fertilizer on a garden. Certain dietary components directly feed specific microbes, helping them grow and thrive.
Like a food source for them.
Exactly. For example, some bacteria in our mouth can take nitrates, which are found in processed meats and some vegetables, and convert them into nitrites and then nitrosamines. These nitrosamines are known carcinogens, directly increasing cancer risk, particularly for stomach and colorectal cancer. So the diet provides the raw material, the bacteria do the conversion.
Okay, so direct fuel for potentially bad actors. What else?
Then there's barrier disruption. Our gut lining is meant to be a strong barrier, keeping microbes and their products mostly contained. But certain dietary deficiencies, like a lack of vitamin D, for instance, can weaken this barrier. It becomes more permeable or leaky.
Leaky gut, I've heard that term.
Right. And when the barrier is leaky, potentially harmful microbes, or even just normal microbes in the wrong place, or their inflammatory products like LPS, can cross into the bloodstream or deeper tissues. This triggers inflammation, and chronic inflammation, as we discussed, is a major driver of cancer development. These potentially harmful commensals that overgrow are sometimes called pathobionts.
Pathobionts. So normally okay, but harmful under the wrong conditions.
Precisely, like when the gut barrier is compromised or the ecosystem is unbalanced. Third, diet causes metabolite modulation. Remember those thousands of chemical signals microbes produce?
The microbial exhaust, yeah.
What you eat dramatically changes the types and amounts of these metabolites. For example, food-derived compounds like taurine found in meat and fish can influence the mucosal immune system, which in turn shapes which microbes can thrive near the gut lining. So diet changes the chemicals, which changes immunity, which changes the microbes. It's all interconnected.
A complex chain reaction.
It really is. And finally, there's direct introduction. Eating fermented foods like yogurt, kefir, kimchi, or taking probiotic supplements can directly introduce new microbes into your system.
So you can just add the good guys?
Well, you can introduce them, but whether they actually take hold and become established members of your gut community, what we call colonization, depends heavily on your existing microbial ecosystem. It's like trying to introduce a new plant species into a dense, established forest. It might not survive or thrive if the conditions aren't right or if there's too much competition. So the effectiveness of probiotics can be quite variable person to person.
Okay, that makes sense. So let's dive into some specific dietary components now, because you said each tells a different story. Let's start with carbohydrates and fiber. Sugar often gets painted as the villain. What's the detailed connection there, linking carbs, cancer, and our microbes?
Right. Carbohydrates are a primary energy source, not just for us, but for our microbes and importantly for cancer cells too. Cancer cells famously exhibit increased glucose uptake. They become incredibly hungry for sugar to fuel their rapid growth and division. This is known as the Warburg effect.
The Warburg effect, like they're glucose addicts.
In a way, yes. They reprogram their metabolism to rely heavily on glucose, and population studies do show correlations. High intake of added sugars is linked with increased risk of certain cancers, like colorectal and breast cancer. Diets high in simple sugars, things that cause a rapid spike in blood sugar, we call this a high glycemic index, are linked to increased risks for colorectal, bladder, and kidney cancers.
So quick energy release foods.
Exactly. White bread, sugary drinks, sweets. Conversely, complex carbohydrates, whole grains, legumes, vegetables which have a lower glycemic index and release sugar more slowly are generally associated with a reduced risk.
What about fructose? That's in fruit, but also high fructose corn syrup.
Good distinction. Fructose, especially the high amounts found in processed foods and sugary drinks via high fructose corn syrup, seems particularly problematic. Studies show it can induce dysbiosis, that imbalance in the gut microbiome we talked about.
Oh.
It tends to reduce the levels of beneficial bacteria that produce butyrate, which we'll talk about more, and increase pro-inflammatory bacteria. This shift might promote tumorigenesis, especially in the context of colorectal cancer. There's also evidence, particularly from animal models of liver cancer, suggesting high fructose intake promotes cancer progression through a complex pathway involving microbial production of acetate, another short-chain fatty acid, which then triggers changes within the liver cells themselves. It's quite intricate.
Okay, so processed fructose seems worse than the fructose naturally present in fruit, which comes with fiber. Let's talk about fiber. It's often hailed as a superfood. How does fiber fit into this complex picture with microbes in cancer?
Fiber is fascinating. Generally, fiber-rich diets show a strong association with lower cancer risk, particularly for colorectal cancer, but also potentially liver cancer. The main reason seems to be its profound effect on gut health.
How does it improve gut health?
Well, firstly, fiber isn't digested by us directly in the small intestine. It travels down to the colon largely intact, where it becomes the primary food source for many of our beneficial gut bacteria. Think bifidobacterium and lactobacillus, types often found in probiotics. Feeding these good bacteria helps them thrive and increases overall microbial diversity, which is generally seen as a marker of a healthy gut.
So fiber is like prebiotic fertilizer for the good guys.
Exactly. That's a great way to put it. And when these bacteria ferment fiber, they produce those incredibly important short-chain fatty acids, or SCFAs we mentioned, mainly acetate, propionate, and butyrate.
Okay, butyrate again. You said it was protective, but sometimes not. Let's unpack that.
Right. Butyrate is probably the most studied SCFA in the context of colon health. It serves as the primary energy source for the cells lining our colon, helping maintain that crucial gut barrier integrity. It also has potent anti-inflammatory effects, for example, by encouraging the development of regulatory T cells, TREGs, which help calm the immune system, and by regulating inflammatory pathways like the NLRP3 inflammasome. So in many contexts, butyrate appears protective against inflammation and the early stages of cancer development.
That sounds really beneficial. So where's the catch?
Here's the twist, the context-dependent effect. Some studies, particularly in specific animal models of established colorectal cancer, have suggested that this same butyrate, under certain conditions, might actually promote tumor growth.
How could it do both?
One proposed mechanism is that it might influence cancer stem cells, potentially making them more resilient or promoting their stemness, which could facilitate tumor progression in an already cancerous environment. It's a concept sometimes called the butyrate paradox.
The butyrate paradox. Protective early on, potentially problematic later.
Potentially in some specific situations. It's a powerful reminder that these microbial metabolites don't have simple good or bad labels. Their effects can depend heavily on the context, the stage of cancer, the specific type of cancer cell, the surrounding microbial community, the host's genetics. It's incredibly complex.
That really underscores the need for personalized approaches down the line.
Yeah.
Okay, so fiber feeds good microbes, produces SCFAs like butyrate, which are mostly good, but have this paradoxical potential. What about fats? They often get a blanket bad rap, but is the story more nuanced, especially concerning our microbes?
Oh, absolutely. The relationship with fats is definitely nuanced. Not all fats are created equal in terms of their impact on cancer risk or the microbiome. High intake of saturated fats, the kind abundant in red meat, butter, fatty processed foods, does tend to promote obesity and chronic inflammation, as we discussed.
And how does it impact the microbes?
It causes significant shifts in the gut microbiome. Typically, high saturated fat diets decrease beneficial bacteria like bacteroids and bifidobacterium and increase the proportion of bacteria that produce lipopolysaccharide, or LPS.
LPS, that the inflammatory molecule you mentioned earlier?
Yes, exactly. LPS is a component of the outer membrane of certain bacteria. When the gut barrier is weakened, which high-fat diets can also contribute to more, LPS can leak into the bloodstream. This condition is sometimes called metabolic endotoxemia.
Endotoxemia, like toxins in the blood.
Sort of, yes. It drives chronic, low-grade inflammation throughout the body, and this systemic inflammation is thought to accelerate tumor progression in various cancers.
Okay, so saturated fats seem problematic via inflammation and LPS.
Mm-hmm.
What about other fats? Unsaturated fats, omega-3s.
That's where the picture changes. Unsaturated fats, particularly omega-3 fatty acids, found in fatty fish, flax seeds, and walnuts, are generally linked to lower cancer incidence in many studies.
How do they work?
The mechanisms are still being explored, but they seem to have anti-inflammatory properties themselves, and they also influence the microbiome. For example, one trial in colorectal cancer patients showed that omega-3 supplementation actually increased the abundance of beneficial SCFA-producing bacteria and decreased pro-inflammatory markers in the body.
Interesting.
And the Mediterranean diet, which is rich in unsaturated fats from olive oil, nuts, and fish, along with plenty of fiber from fruits, vegetables, and legumes, is consistently associated with decreased inflammatory markers and better cancer prevention outcomes. Its beneficial effects are thought to be mediated, at least in part, through its positive impact on the gut microbiome.
So for fats, it's really about the type of fat, saturated fats, potentially driving inflammation via LPS and microbial shifts, while unsaturated fats, like omega-3s and those in a Mediterranean pattern, seem beneficial. What about proteins? We hear a lot about red meat and cancer risk. Let's get into the specifics there, including how microbes might play a role.
Right. Proteins are essential, but the source matters significantly for cancer risk. Red meat, and especially processed meats like bacon, sausage, ham, are consistently associated with a higher risk of colorectal cancer. In fact, processed meat is classified by the World Health Organization as a Class 1 carcinogen.
Class 1. Same category as smoking.
In terms of the strength of evidence linking it to cancer, yes, though the magnitude of risk is different. One major reason is that when these meats are cooked, especially at high temperatures, carcinogenic compounds called heterocyclic amines, HCAs, and polycyclic aromatic hydrocarbons, PAHs, can form.
Okay, HCAs and PAHs, and the microbiome interacts with these.
Potentially, yes. There's some intriguing research suggesting that certain beneficial gut bacteria, like some lactobacillus strains, might actually help inhibit the DNA damage caused by these HEAs, perhaps by binding to them or metabolizing them into less harmful forms. It's an area of active research. Can microbes offer some protection?
That would be amazing if they can mitigate some harm. What else about red meat?
Another factor is the heme iron content in red meat. While iron is essential, high levels of heme iron in the colon can be problematic. It can generate oxidative stress and contribute to gut dysbiosis imbalances in the microbiome. Studies suggest it can reduce overall microbial diversity and promote the formation of adenomas, which are precancerous polyps in the colon.
Interesting. So heme iron itself is a factor.
Yes, and microbes are involved there too. Some research indicates that specific types of bacteria, particularly those that produce hydrogen sulfide or degrade the protective mucus layer in the gut, like Akkermansia in some contexts, might be involved in mediating the harmful effects of heme. Interestingly, in one animal study, giving antibiotics actually abrogated the hyperproliferation, the excessive cell growth induced by heme in the gut lining, presumably by eliminating these key bacteria.
Wow, another complex microbial role. So what about better protein sources?
Plant-based proteins from legumes, nuts, seeds, tofu, and fish are generally linked to lower cancer risks compared to red and processed meats. They don't have the heme iron issue, don't typically form HCAs to the same extent, and often come packaged with fiber and other beneficial compounds.
Okay, and what about individual amino acids, the building blocks of protein? Do they matter?
They do. It's a very intricate level of detail. Tumors have unique metabolic needs. For example, many tumors are addicted not just to glucose, but also to certain amino acids like glutamine and branched-chain amino acids, BCAAs, leucine, isoleucine, and valine. They use these as alternative fuel sources and building blocks. This has led to research into restricting these specific amino acids in the diet as a potential therapeutic strategy.
Starving the tumor of its preferred fuel.
That's the idea. And then there's tryptophan. Its metabolism is heavily influenced by the gut microbiome, and the end products can be wildly different. Some gut microbes, like Morganella morganii, can convert tryptophan into derivatives that might be genotoxic, potentially promoting inflammation in cancer, especially in conditions like inflammatory bowel disease, IBD.
So tryptophan breakdown can be bad.
It can be. But other microbes, like Lactobacillus reuteri, can convert tryptophan into different compounds, like indole-3-aldehyde, which can actually be beneficial. This compound can signal through the immune system and has even been shown to enhance the effectiveness of PD-1 blockade immunotherapy in melanoma models.
Amazing. The same starting amino acid, tryptophan, but depending on which microbes are acting on it, you get completely different outcomes for cancer and treatment.
Exactly. It perfectly illustrates how the microbiome acts as a crucial chemical processing plant for our diet with profound downstream effects.
So many intricate interactions. Okay, what about the smaller players? Minerals, vitamins, and all those modern additives crammed into processed foods. Are they influencing our microbes and cancer risk too?
Absolutely. Fruits and vegetables, of course, provide essential vitamins and phytochemicals, many with antioxidant and cancer-preventive properties. Minerals like selenium and zinc also play important antioxidant roles. However, it's worth noting that excessive supplementation with certain vitamins has sometimes shown mixed or even potentially harmful results in large trials, so getting them from whole foods is generally preferred.
Right, the balance is key. How do minerals interact with microbes?
Dietary minerals definitely interact. We mentioned heme iron, but even supplemental iron or manganese can preferentially feed certain potentially harmful bacteria or pathobionts, like some strains of Escherichia coli. Promoting their growth can indirectly contribute to cancer through inflammation and oxidative stress.
So even essential minerals can be problematic as they feed the wrong microbes.
Potentially, yes, especially in supplemental forms or high amounts. And consider salt. A high-salt diet shows really contradictory effects in cancer studies. In some models, like mammary and melanoma tumors, it surprisingly inhibited tumor growth, possibly by increasing gut permeability in a specific way that allowed immune cells, like NK cells, to be activated against the tumor.
Inhibited growth, that's unexpected for high salt.
Very. But in liver cancer models, a high-salt diet seemed to exacerbate fibrosis and inflammation, potentially by promoting the translocation or leakage of specific gut bacteria like Enterococcus faecalis from the gut to the liver.
Wow. Completely opposite effects depending on the cancer type and context.
It really highlights the complexity.
Hmm.
And then you have the additives in modern processed foods. This raises a really important question about our modern food environment.
Yeah, emulsifiers, artificial sweeteners. What's the story there?
Synthetic additives like emulsifiers, things like carboxymethylcellulose, CMC, and polysorbate 80, often found in ice cream, baked goods, salad dressings to improve texture, and non-caloric artificial sweeteners, NAS, like sucralose or aspartame, are increasingly scrutinized. Animal studies suggest these can induce gut dysbiosis, disrupt the gut barrier, promote low-grade inflammation, and potentially alter metabolism in ways that could impact cancer risk, perhaps by promoting obesity or metabolic syndrome. However, direct links to cancer in humans are still mostly hypothetical and require much more research. Many of these compounds are relatively new in our evolutionary diet, and their long-term impacts, especially on the microbiome and cancer, are not fully understood.
So a lot of unknowns with those additives. When we look at whole diets, not just single nutrients, what patterns emerge?
It's definitely hard to isolate individual nutrients because people eat foods in combination. That's why studying overall dietary patterns can be more informative. Broadly, we see patterns. Plant-heavy diets, rich in fiber and diverse phytochemicals, tend to enhance microbial diversity and promote bacteria that produce beneficial SCFAs. Diets high in animal products, particularly red meat, tend to promote bile-tolerant bacteria and those involved in pathways that might be pro-inflammatory or produce potentially harmful metabolites like hydrogen sulfide. And those high-fat, high-sugar Western diets consistently shift microbial activity toward sugar and fat metabolism, often contributing to inflammation and metabolic dysfunction.
Okay, that was an incredibly detailed journey through how specific food components interact with our microbes to influence cancer risk. Let's try to consolidate that. We saw carbs and sugars fueling cancer cells directly, but also altering microbes, with processed fructose being particularly concerning. Fiber feeds beneficial microbes producing SCFAs like butyrate, which is mostly protective but has that complex paradox. Fats depend on type-saturated fats driving inflammation via LPS, unsaturated fats often beneficial. Proteins also depend on source red processed meats linked to risk via HCAs and Hymes, while plant proteins fish seem safer, and even amino acid metabolism, like tryptophans, is microbe-dependent with huge implications. Minerals and additives add more layers, sometimes with paradoxical effects or unknowns. So the big picture is, nearly everything we eat is constantly shaping this internal ecosystem, and those changes have real consequences for inflammation, metabolism, DNA damage, and ultimately, cancer risk. Thinking about your own diet, which of these connections feels most relevant or surprising to you? We've covered what we eat in incredible detail, but the outline mentions another factor, when we eat. This whole concept of dietary timing and circadian rhythms, it sounds like adding another dimension of complexity.
It absolutely is another crucial dimension. Most living organisms, including us and our microbes, have evolved internal biological clocks that regulate our physiology and behavior over a roughly 24-hour cycle. These are our circadian rhythms.
Our body clocks.
Exactly. And these rhythms govern sleep-wake cycles, hormone release, metabolism, immune function, and they're tightly linked to natural cycles of light and dark, and importantly, cycles of feeding and fasting.
So disrupting these rhythms could be bad for health.
Yes, we see this quite clearly in studies of long-term shift workers, for example, whose schedules constantly disrupt their natural circadian rhythms. They have increased risks for various chronic diseases, including metabolic syndrome, cardiovascular disease, and importantly, several types of cancer, including colorectal, prostate, and breast cancers.
Just from the timing disruption.
It seems to be a significant contributing factor, yes. The chronic misalignment between their internal clocks and their external environment, including eating patterns, appears to create physiological stress and dysregulation that can promote disease.
And you mentioned our microbes. They have their own clocks, too. It's like a whole city inside us running on schedules.
That's a great analogy. Yes, the gut microbiome isn't static throughout the day. It undergoes its own significant circadian oscillations in both its composition, which microbes are active, and its function, what metabolites they're producing.
And what drives the microbes' clocks?
It seems to be a combination of factors. They respond to our own host circadian signals, like hormones released on a 24-hour cycle. But they're also profoundly influenced by our feeding times. When we eat provides a major timing cue for the gut ecosystem.
So what happens if we disrupt that microbial rhythm, like with jet lag or shift work eating patterns?
Disrupting the host's rhythm through things like experimental jet lag or altered feeding schedules in animal models significantly throws off the microbial diurnal activity as well. You lose those regular oscillations, and this microbial disruption, in turn, signals back to the host, impacting our metabolism, our immunity, potentially contributing to the health problems associated with circadian disruption. It's truly a two-way street. Our clock influences the microbes, and the microbial clock influences us.
Okay, this is fascinating. What are the practical implications of understanding this circadian microbiome cancer axis? Could something as simple as when we eat become part of cancer prevention or even treatment?
The research here is still relatively nascent compared to, say, specific nutrient research, but it's incredibly promising and growing fast. We're already seeing hints of its potential. For instance, studies involving caloric restriction, CR, or periodic fasting, which inherently involve changes in eating timing, have shown they can improve therapy outcomes and quality of life for some patients undergoing chemotherapy, for example, for breast and ovarian cancers.
How does fasting or CR help?
Part of the benefit seems to come from reshaping the gut microbiome. CR can promote the growth of certain beneficial bacteria and alter metabolite production in ways that generate anti-tumoral effects or ameliorate adverse side effects of treatment. For example, one study showed that enrichment of an acetate-producing bacterium, Bifidobacterium bifidum, under CR conditions increased resistance against colorectal cancer formation in mice.
Interesting. So changing the timing and amount of food changes the microbes, which helps fight the cancer or side effects.
That seems to be a key part of the mechanism, and this has led to the development and testing of more structured periodic dieting approaches like time-restricted eating, TRE, and fasting mimicking diets, FMDs.
Okay, TRE and FMDs. Let's break those down. What's time-restricted eating?
TRE isn't necessarily about what you eat or how much overall, but consolidating your eating into a specific window each day, typically 8-12 hours, and fasting for the remaining 12-16 hours. The idea is to reinforce the body's natural circadian rhythms by having consistent daily periods of feeding and fasting.
What are the potential benefits?
Studies, mostly in animals and some human trials, suggest TRE can lead to weight loss, improve insulin sensitivity, reduce markers of inflammation, and potentially improve cardiovascular health. In the context of cancer, some epidemiological studies hint at associations between TRE patterns and lower risks for prostate and breast cancer. And in mouse models, TRE has been shown to reduce lung tumor growth, partly by altering gut commensal bacteria.
So reinforcing that daily rhythm might be protective. And fasting mimicking diets, FMDs. They sound intriguing, like getting fasting benefits without the full fast.
Exactly. FMDs were designed to capture some of the metabolic and cellular benefits of prolonged fasting, but in a way that's generally safer and much easier for people to stick to. They typically involve significantly reducing calorie intake for several consecutive days, maybe 3 to 5 days, following a specific macronutrient composition relatively high in healthy fats, but low in protein and sugar.
And the cancer connection.
Animal models are quite compelling here. FMD cycles have been shown to decrease spontaneous tumor formation and hinder the development of existing tumors. When combined with conventional therapies like anti-estrogen treatment for breast cancer, FMDs led to higher rates of tumor remission in mice. This seems linked to FMDs lowering levels of growth-promoting factors like insulin, leptin, and IGF-1.
Are these being tested in humans?
Yes, early-stage human trials are underway. They suggest that FMD cycles are generally safe for cancer patients and can induce beneficial metabolic changes, potentially enhancing anti-tumor immunity. It's still early days for clinical application, but the potential to use FMDs alongside standard therapies is generating a lot of excitement.
Okay, let's summarize this timing aspect. We've moved beyond just what we eat to exploring when we eat. Our bodies and our microbes run on internal 24-hour circadian clocks. Disrupting these clocks, through things like shift work or erratic eating, is linked to increased cancer risk. Conversely, strategies that align with or leverage these rhythms, like caloric restriction, time-restricted eating, confining eating to a daily window, or fasting mimicking diets, periodic low-calorie cycles, show real promise. They seem to work partly by reshaping the gut microbiome, improving metabolism, reducing inflammation, and potentially enhancing cancer treatment efficacy or reducing side effects. Does learning about this make you reconsider your own daily eating schedule? This next part is truly where the science gets incredibly exciting, I think. How diet and the microbiome can directly influence the effectiveness and side effects of actual cancer therapies. Feels like a completely new frontier in treatment personalization.
It absolutely is. We're seeing diverse and sometimes surprising impacts on whether a therapy works well, sensitivity or doesn't, resistance, and also on a patient's likelihood of experiencing severe adverse effects. Think of it like using diet and microbes to fine-tune the body's response to powerful medications.
So let's go through the different types of therapy. How does this play out with chemotherapy?
Several connections are emerging. For instance, preclinical studies show that dietary intake of the amino acid histidine can enhance sensitivity to the chemo drug methotrexate. We also touched on fasting cycles. These have been shown, again, mainly in preclinical models so far, to not only reduce tumor growth, but also sensitize various cancer cells to chemotherapy drugs like doxorubicin, potentially allowing for lower doses or better effects.
And the microbiome's role in chemo response.
It's crucial here, too. Remember tryptophan metabolism. One specific metabolite derived from tryptophan by gut bacteria, called 3-IAA, indole-3 acetic acid, produced by bugs like Bacteroides fragilis, has been associated with better responsiveness to certain types of chemotherapy in pancreatic cancer patients.
So having the right microbial metabolite could predict chemo success.
It might be a biomarker, yes. And butyrate, that SCFA we keep coming back to, can sometimes augment the effects of certain chemotherapy drugs in colorectal cancer and other cancers, possibly by altering cancer cell epigenetics or metabolism. Ketogenic diets are also being studied for their potential to enhance chemotherapy response in some contexts, maybe by altering tumor metabolism.
Okay, so diet composition, timing like fasting, and specific microbial metabolites are all interacting with chemotherapy. What about radiotherapy?
Similar themes emerge. Some studies suggest that structured dietary counseling during radiotherapy can improve patient responses and outcomes. The ketogenic diet, again, has shown potential in preclinical models to enhance radiochemotherapy responses, possibly by increasing oxidative stress specifically within the tumor cells, making them more vulnerable to radiation damage.
Does radiation affect the microbiome?
Oh, yes, definitely. Radiotherapy, especially to the abdominal or pelvic area, significantly alters the gut microbiome composition. And intriguingly, the microbiome seems to talk back. Some research suggests certain gut bacteria, specifically ones sensitive to the antibiotic vancomycin, can actually enhance the anti-tumor activity of radiotherapy.
Bacteria helping radiation work better.
It seems so. But here's another layer of complexity. That beneficial effect could be abrogated or canceled out by butyrate.
Butyrate again. So it helps chemo sometimes, but might hinder radiotherapy's microbial boost.
In this specific context, that's what the preclinical data suggested. It really underscores how these interactions are highly context-dependent. What's beneficial in one scenario might not be, or could even be detrimental in another.
Complexity is the word of the day. Okay, what about surgery? Does the microbiome influence recovery after cancer surgery?
Yes, this is a growing area of interest. The preoperative state of the gut microbiome seems to impact post-surgical healing, particularly after bowel surgery for colorectal cancer. For example, anastomotic leakage, where the reconnected bowel doesn't heal properly and leaks, is a serious complication.
Right.
One study found that mice receiving FMT from human CRC patients who unfortunately suffered anastomotic leakage exhibited poor healing themselves compared to mice getting FMT from patients who healed well. This suggests the microbiome plays a role in tissue repair after surgery.
And diet leading up to surgery.
It seems to matter. A typical Western diet, high-fat, low-fiber, was shown in mice to promote intestinal colonization by microbes that produce collagenases, enzymes that break down connective tissue. This actually enhanced tumor recurrence after colorectal surgery in these models.
Wow, the diet promoted microbes that interfered with healing and helped the tumor return.
Exactly. But the good news was that switching these mice to a standard chow diet with fiber before surgery helped improve healing outcomes. It suggests preoperative dietary modulation could be a strategy to improve surgical recovery.
That's really practical potential. Now, the big one, immunotherapy. We hear so much about it. How do diet and microbes influence these cutting-edge treatments?
This is perhaps where the diet microbiome link is most dramatically evident right now. The gut microbiome can significantly modulate patient responses to immune checkpoint blockade, ICB drugs like PD-1 or CTLA-4 inhibitors.
Modulate how? Better or worse?
Both. Studies consistently show that depleting the gut microbiome, for instance with broad-spectrum antibiotics, is often linked to worsened overall survival for patients receiving ICB therapy. Having a diverse and favorable microbiome seems crucial for these drugs to work effectively.
So antibiotics could potentially sabotage immunotherapy.
That's a major concern, yes, and why oncologists are increasingly careful about antibiotic use in patients starting immunotherapy. On the flip side, the potential for positive modulation is huge. We mentioned FMT earlier in pilot studies. Transferring fecal microbes from melanoma patients who responded well to immunotherapy into patients who didn't respond initially actually induced partial or even complete responses in about 30% of those non-responders.
That's incredible, changing the gut microbes turn non-responders into responders.
It's truly groundbreaking, though still experimental. And diet plays a role here too. Remember the tryptophan metabolite produced by Lactobacillus reuteri? A tryptophan-enriched diet was shown to enhance PD-1 blockade therapy in mice, likely via this microbial pathway boosting immune function within the tumor.
So diet could potentially prime the microbiome for immunotherapy.
That's the hope. Even seemingly unrelated things like a high-salt diet showed surprising pro-immunotherapy effects in some preclinical models, possibly by enhancing T-cell and NK cell functions, though translating this to humans needs extreme caution. And dietary timing comes in again. Fasting and FMDs have also been shown in animal models to improve ICB efficacy and reduce immune-related adverse effects in breast and lung cancers.
So many potential levers to pull. What about those tough side effects of treatment generally? Because reducing suffering during cancer therapy would be a massive win. Can the microbiome help there too?
Yes, very importantly, it can. Many cancer therapies, particularly chemotherapy and radiotherapy to the gut area, cause severe gastrointestinal toxicities. Mucositis, which is painful inflammation and ulceration of the lining of the digestive tract, and severe diarrhea are common and debilitating side effects.
Can microbes predict or prevent these?
There's evidence for both. Microbiome profiling before treatment might help predict who is at higher risk for severe side effects, like radiation-induced esophagitis. And certain beneficial commensal bacteria seem protective. For example, bacteria belonging to the Bacteroidetes phylum have been shown to protect against CTLA-4-induced colitis, inflammation of the colon, a common immunotherapy side effect. Another specific bacterium, Faecalibacterium prausnitzii, known for its anti-inflammatory properties, seems to suppress immunotherapy-associated intestinal toxicity.
So having these good bugs could shield patients from some side effects. Can we boost them with diet?
That's the goal of dietary interventions. For example, supplementing the diet with specific types of fiber to enrich SCFA-producing bacteria, or even directly supplementing with butyrate, has shown promise in preclinical models for protecting against chemotherapy-associated mucositis and gut barrier impairment. Dietary timing, like fasting, has also been shown to reduce intestinal damage from radiation in animal studies.
So manipulating diet microbes isn't just about making treatments work better, but also making them more tolerable.
Exactly. Improving quality of life during treatment is a huge unmet need. However, we have to circle back to that complexity issue. As we noted, some compounds like butyrate might favorably modulate chemotherapy effects but could adversely impact radiotherapy responses or correlate with poor immunotherapy outcomes in certain contexts. It's not straightforward.
Right. Okay. Another big recap then. Diet and the microbiome are emerging as critical modulators of virtually all major cancer therapies: chemo, radiation, surgery, and especially immunotherapy. Specific nutrients, dietary patterns like keto or fasting FMDs, and individual microbial species or their metabolites can influence treatment sensitivity, resistance, and crucially, side effects like gut toxicity. FMT shows incredible promise, but also highlights the power of the existing microbiome. But the effects are highly context-dependent, with things like butyrate showing different effects depending on the therapy. What does this complexity tell you about the future need for truly personalized treatment plans? This entire field is clearly buzzing with potential. It's so exciting, but it's also, as you keep emphasizing, incredibly complex.
Yeah.
What are the biggest hurdles we need to overcome to actually translate all this groundbreaking research into real, everyday patient care, into standard practice?
That's the critical question, isn't it? A major, perhaps the biggest challenge is the relative lack of definitive evidence-based proof of causality for many of these links, especially in humans.
Still stuck on correlation versus causation for many things.
For many things, yes. Current clinical guidelines from major bodies like the American Society of Clinical Oncology, ASCO, or the European Society for Clinical Nutrition and Metabolism, ESPEN, offer general healthy eating advice, but they don't recommend specific diets like ketogenic or high fiber for cancer patients as a standard treatment component.
Why not, if the research looks promising?
Primarily because much of the data, especially human data, remains correlational or comes from preclinical models. We need more large-scale, well-designed, randomized controlled trials in humans to establish clear cause and effect relationships before we can confidently change clinical guidelines. These trials are expensive, difficult to conduct, and take a long time.
What makes them so difficult?
Well, there are numerous methodological limitations. In animal models, dietary protocols can be inconsistent between labs, and sometimes the interventions used are quite extreme, like zero fiber or incredibly high fat, which might not accurately reflect typical human diets.
Right.
In human studies, we face huge challenges with things like selection bias. Are the people who volunteer for diet studies different from the general population? Small cohort sizes, short follow-up times, and perhaps the biggest one, compliance and accurate dietary assessment.
Measuring what people actually eat. That sounds tough.
It's notoriously difficult. Traditional methods like food diaries or recall questionnaires rely on people accurately remembering and reporting everything they ate, which is prone to errors and biases. This raises a really important question. How do we accurately measure what people are truly eating and how their bodies are processing it in real time?
There are new ways.
Yes, thankfully. New technologies are emerging. Things like genome-based and proteome-based pipelines are being developed to try and get objective measures of the dietary exposome, basically, quantifying the actual dietary compounds and microbial metabolites present in someone's blood or urine. This could give us a much more accurate picture than just asking people what they ate.
Objective biomarkers instead of self-reports. That sounds promising. What about the fact that everyone is so different?
Exactly. Host heterogeneity is another massive challenge.
Sure.
Our individual genetics, our baseline microbiome structure, which is unique as a fingerprint, even factors like how quickly food moves through our gut transit time, all these things can lead to vastly different responses to the same dietary intervention.
So my response to a high-fiber diet might be totally different from yours.
Precisely. Decoding this incredible individual variability requires analyzing huge data sets, big data, integrating dietary information, microbiome data, host genetics, clinical outcomes. And this is where artificial intelligence and machine learning applications are becoming invaluable.
How is AI helping?
AI algorithms are already proving useful in predicting personalized glycemic responses to different foods based on microbiome data, for instance. The hope is that similar AI approaches can help us predict which dietary or microbial interventions will be most effective for a specific cancer patient based on their unique profile. It's the foundation for true personalization.
What about things people are already trying, like grabbing probiotics off the shelf? Do they fit into this future picture?
That's a common question. Standard over-the-counter probiotics often suffer from those conflicting results we talked about, partly because of variable colonization resistance. Your existing gut microbes might simply prevent the probiotic strains from taking hold.
So they might just pass right through.
Often, yes, or only have transient effects. The future here likely lies in what's being called precision probiotics, or maybe next-generation probiotics. These would involve carefully selected, often gut-resident strains, maybe even genetically engineered strains, specifically chosen for their ability to effectively colonize a particular individual's gut and exert a desired, predictable effect, perhaps producing a specific metabolite or modulating the immune system in a certain way.
Tailor-made microbes.
Essentially, yes. Ultimately, the big leap we need to make is moving beyond just observing associations and correlations towards understanding the precise causal links and the underlying mechanisms. How exactly does this bacterium or this metabolite influence that cancer pathway?
How do we figure out those mechanisms?
We need sophisticated experimental platforms. Germ-free animal models are crucial for studying the direct effects of specific microbes. We also need better in vitro systems, like advanced gut simulating systems, gut on a chip, organoids, mini organs grown in the lab, which allow us to study these interactions in a controlled, human-relevant context. We also desperately need to diversify research beyond the typical developed world populations to understand global variations in diet, microbiomes, and cancer patterns. And critically, researchers need to consistently report crucial metadata in their studies, like recent antibiotic use, which can profoundly impact the microbiome and confound results if it's not accounted for.
So pulling all that together, what's the ultimate vision for the future? How will this deep dive into diet and microbes truly change personalized cancer care down the road?
The ultimate goal is to zoom in with precision on those specific bacteria, fungi, or viruses and their bioactive products or metabolites that have been causally established as promoting or inhibiting cancer or influencing treatment response.
Once we know the key players and what they do,
Then we can develop truly precision dietary approaches, not just general advice, but tailored nutritional strategies designed to optimize beneficial microbial functions and suppress harmful ones for that specific patient. And we can integrate these dietary strategies with other emerging microbiome-targeted therapies.
like the precision probiotics you mentioned.
Exactly. Precision probiotics, perhaps metabolite supplementation, directly giving beneficial microbial products like specific SCFAs or indole derivatives, sometimes called postbiotic therapy, or maybe even targeted phage therapy using bacteriophages, which are viruses that selectively infect and kill only specific types of bacteria, to precisely remove harmful microbes from the gut without broad-spectrum antibiotics. The vision is to create a truly holistic, personalized cancer care paradigm where nutrition and microbial modulation are integral, data-driven components alongside surgery, radiation, chemotherapy, and immunotherapy.
Empowering patients with these highly specific, personalized strategies based on their own biology and microbiome.
That's the future we're working towards.
Wow. We have journeyed through an incredible, often hidden world today. The intricate intersection of your diet, your gut microbes, and cancer. From digging into why something like a Western diet contributes to cancer risk, partly through inflammation and microbial shifts, to understanding how specific bacteria within you might actually enhance or hinder treatments like chemotherapy or immunotherapy. It's just become so clear that our daily choices are constantly influencing this complex internal dialogue inside us.
It is incredibly complex. The sheer number of interactions is daunting, no doubt. But the potential here for genuinely transforming cancer prevention strategies, improving treatment efficacy, and reducing side effects is just immense. The key, as we discussed, is rigorously moving from correlation to establishing causation. That knowledge will unlock truly personalized nutritional and microbial interventions.
It really makes you think, doesn't it? Every single meal you choose could be a profound interaction with your unique internal ecosystem, influencing your health trajectory in ways we are only just beginning to truly grasp. How might knowing this change your daily choices? What big questions does this spark for you about the future of personalized medicine and how we manage health? We hope this deep dive today has given you a powerful, maybe even slightly overwhelming, but definitely fascinating new lens through which to view your health, your diet, and that bustling microscopic world within.